All right, everybody, it is chapter 12 time. We are moving on to the brain, and the cranial information here is going to piggyback onto what we've done in lab so far. So, like we spoke in lab, there are 4 parts to the brain. The one that normally comes to mind is the cerebrum, and those have like the 4 lobes of our brain, the limbic, the insula, the corpus callosum, all those big features, and the sulci and the gyri mixed in. But that's not the only part. The next biggest part is the cerebellum, which sits on the back lower portion and plays a big role in coordination and trying to even out our movements, so when we try to grab for a drink, we don't overshoot or we don't go too much to the right. You know, it's kind of like it helps us, if we were a kicker, it helps make sure we always get through the uprights and we don't miss right, miss left, short, or overdo it, you know. Anyway. The diencephalon is where the 2 thalamus are located, those 2 big areas on each hemisphere, and then in the center on the midline is going to be our hypothalamus region. We also associate the pineal and our pituitary glands to the hypothalamus as well. The brainstem is going to be the most oldest part of the brain, the part that, if you look at for homology purposes, relates back to early areas of worms and then squid and all the things you do in zoology. The brainstem is divided into 3 parts: the midbrain, the pons, and the medulla oblongata. And there are some controls within these parts that are all subconscious. So this is where a lot of our autonomic nervous system comes out, controlling our breathing rate, controlling our heart rate, controlling some of those little nuances that we don't need to be thinking about day in and day out in conscious awareness. Okay? Now, what's also important is within the cerebrum in the brain are fluid-filled sacs called ventricles. And the reason why we have these fluid-filled sacs along with the meninges with a fluid-filled subarachnoid space is this allows the brain to be completely floating in a water matrix. And so the brain then is better for cushioned, better for shock absorption, and so every time you don't move your head or you shake your head, your brain tissue and the meninges are not pushing and slamming or having damage, micro damage, as it hits or bumps into the skull, the bones. So that water is important because it, one, brings nutrients over, because we're going to see that the blood supply doesn't go all the way to every single neuronal cell like it did in our skeletal muscle. And so we need a way to get fluid and glucose and sodium and nutrients to the neurons and pull out their waste products. But we also want to keep the brain tissue as light as possible and suspended in a fluid to better help it be protected from the elements. All right, so let's start with the cerebrum, the big part. This is the part that makes humans, you know, humans in some ways, that we have such a large cerebrum. We've grown into these big lobe areas that have higher mental function. So we are the capacity for emotions, for logic, for mathematics, for language. They're divided into a right and left hemisphere. And when you go through psychology class, hopefully you'll go into more of personality and emotions and how that is all tied to the right or left brain hemisphere. So looking at white versus gray matter, where are the myelinated axons that live in the brain and where are most of the cell bodies and synapses occurring? Most of the gray matter is going to be on the outside of the brain. So it's going to be in the cortex, the outer kind of quarter to a third part of brain matter. So if I was to take my fingernails and scratch, I would be scratching on these gyri here, I would be scratching gray matter. Now, are there little pockets of gray matter embedded in the white matter more inside the brain? Yes, and those we're going to call ganglia areas, nuclei, but most of the white matter is going to be on the middle or inside of the tissue. So if I wanted to get to the white matter, the myelinated axons of this region, I would need to dig into this lobe a few inches or a few centimeters to be able to find where the axons that are myelinated are coming off of these cell bodies and heading to their next destination. So because of the way the gray and the white matter are arranged, you can call the outermost part of the cerebrum the neural cortex. It's the area where cell bodies are going to begin and end all of our descending and ascending pathways. So this is where those neurons basically bring sensory information to a focus of this is our perception of reality. These are the sounds, the smells, and all that stuff comes into play. This is where motor function originates. So this is where the original cell body neuron that is going to start the pathway that ultimately leads to me wiggling my fingers or moving my lips. Okay? Again, to make as much space as possible for the neurons, we don't just want the brain to be like a loaf of bread. We want it to have more potential rivets and divots to it. And so that's why you have folded surfaces where the elevated, kind of like the meaty part of the tissue is the gyri, and where the two gyri kind of push into each other. They're technically physically separated, but they're just squeezing into the space and therefore kind of squishing next to each other. That's going to form a depression or a ditch that we call the sulci. Major divisions that you see a little more pronouncedly, like between the two hemispheres, they're still deep grooves and depressions, but we'll call them fissures, right? And so for the lobes are going to get their name by what bone they usually are closest to. So the very front of the brain is closest to the frontal bone in your forehead, so thus it's the frontal lobe. There is usually a very distinctive sulcus that runs 90 degrees to our longitudinal hemisphere. That's your central sulcus, and that's where typically the frontal lobe ends and the parietal lobe underneath the parietal bone begins. It's not as clear, but the occipital lobe is going to be the very back cerebrum part of the brain, and that's part of the reason why when people bump the back of their head, sometimes they see stars or they for a few seconds kind of lose visual ability, because that is the area of the brain, and the outermost part of that area is the occipital cortex, which is where visual information from your eyes is heading to be made into your visual field of view. The temporal lobe is going to be this area out here, closest to the eyes and closest to the temporal bone. All right? So that's the four major lobes that you can see from an external view. The insular or the limbic lobe, the limbic system, it depends on the book which you read, can sometimes be called it's the fifth lobe. Sometimes it's just a combination of little bits of each lobe right on top of the corpus callosum, the white matter that connects the two hemispheres, and it's called the limbic system. So limbic lobe, limbic system, it really depends on the book if what they refer to it as. Okay. Now, underneath the cerebrum is this white matter area. So this is the corpus callosum, and you can see the corpus callosum is a bunch of white myelinated axons. So a lot of oligodendrocytes around, lots of axons here that are connecting the cerebrum and the other regions of the brain on the right hemisphere to the cerebrum and the other hemisphere of the left brain. All right? Underneath, and it kind of, if you think of this as like the eyeball, and then this is like the hair or the feathers, it kind of, and then this is like the beak, and then like a chicken or a turkey. It kind of looks like a bird somehow. I don't know, maybe just to me. But you got to think three-dimensionally. This is on midline. So the area that's the hypothalamus, there's a little bit of hypothalamus here and a little bit of hypothalamus on the other half. But the thalamus, this is the like medial border of the thalamus that then shoots and becomes part of that hemisphere, that direction, and then there's another one on the other side. All right? Think of the thalamus as a nice gate guard before you go into a really expensive neighborhood. All right? So the cerebrum is a really expensive neighborhood. There's a lot of high-functioning, rich things going on here. And so before things are allowed to leave and before things are allowed to come in, the thalamus is an area where a lot of the neurons that are cell bodies in the cortex, axons travel to the thalamus and end, and they terminate. And then at the hypothalamus, there's a cell body of another axon that then will continue down into the spinal cord. And so the thalamus is an area where there's some gray matter here where axons are ending and cell bodies reside, and they're part of these ascending and descending pathways for motor control and sensory information. So when you read and you get into kinesiology and OTA and PTA and you start thinking about, okay, there's sensory dysfunction in the dorsal medulla pathway, well, you'll figure out that that pathway might have 3 neurons total. And it might be that the second neuron in the pathway and the final third neuron, which is going to end in the cortex of the brain, synapse and communicate to each other in the thalamus, and the vice versa on some of the descending pathways. So the thalamus is kind of, again, a nice gatekeeper, a nice point where before things leave, they do this stop, they have the synapse, and then another neuron takes the message onward. And before things come in, again, they end, they synapse in the thalamus, and then another neuron takes it to the table, to the final location. Remember that the hypothalamus is a big control place. It controls a lot of our neuro functions of temperature, of appetite, circadian rhythm, but it also plays a role in endocrine function with control of water balance, with ADH, with some smooth muscle contractions and oxytocin, and then all of the releasing and inhibitory factors that it controls, FSH, LH, from the pituitary gland. The pineal gland is back here. And again, the pineal gland is going to release melatonin. The pineal gland is going to have a somewhat an ability to interact with the optic nerve and the light impulses going towards the occipital lobe. And that melatonin is going to feed back along with other triggers with the hypothalamus to help us set our circadian rhythm, our day cycles, our night cycles, our seasonal cycles. Okay? So looking at the hypothalamus, we've mentioned that it's a big set point for a lot of things going on in our body. So it's where we provide subconscious control of skeletal muscle. We control a lot of autonomic function. This is where there's a lot of control coming up for just, again, day-night cycles and some of the changes that come with that. We control nervous as well as endocrine systems. We secrete a lot of hormones. We take in a lot of information related to food and nutritional balance and hunger and satiety. We do body temperature. We do, again, circadian cycles. And now the hypothalamus, again, they're talking about nuclei. Certain areas of it are called nuclei. So again, it's going to have these little gray pocket areas where a lot of maybe cell bodies are located, communicating with each other, synapsing with each other. They're unmyelinated, so they're gray areas, or some other axons are coming in, some blood supply is coming in. And so it's an area with not a lot of myelinated axons, and those little pockets and areas form these little gray spots, little gray nuclei, and that's why they're called nucleus. Okay? So they're not really a nucleus of a cell, they're just these big gaggles of neurons and the non-myelinated parts of neurons and communicating synapses between neurons that then collectively tend to play a big role in controlling certain outputs of either hormones or neurons or neurotransmitters, or controlling certain cycles or parts of whatever the hypothalamus does. Okay. Moving to the third part of our brain, which is the brainstem. And again, the brainstem is the diencephalon, which is the most superior part. I'm sorry, the diencephalon sits on top of the most superior part of the brainstem, which is the midbrain. Right? So this green part is the midbrain. And we don't know a lot about the midbrain. It's the midbrain because it's very hard to put probes on it. It's very hard to get to it. It's very hard to find it because it's embedded somewhat deep in the middle part of our brain. And we know that for most of what's going on, there's some reflexes built into the midbrain that are survival type, reflexes. So if I hear a loud sound or a crunch of leaves, like any animal, that would startle you, that sound, so you would turn and look at it. So that reflex of I hear a sound, turn and look at it, is one of the reflexes that we know kind of tie into the midbrain. And again, the whole purpose is to, if something is coming towards us and we don't know what it is, let's look at it and determine if we're going to run away or ignore it. The pons is going to relay information to the cerebellum. So there are going to be axons that run right, left, basically front, back to the cerebellum. And there'll be axons that run north-south that continue upwards to the thalamus eventually, where it can then synapse and then continue on or continue down. But other regions of the pons that are going to be where these little gray area axons and, cell bodies are going to play a role in some higher modulation on the medulla for breathing and some higher modulation of other somatic and visceral motor centers, okay? The medulla oblongata is the most archaic part of the brain, and we know for sure there are three key areas. There's a breathing area, so we know that when we go to sleep, as long as we have an intact medulla, there's an area in there where the axons are going to trigger a contraction of the diaphragm on a consistent basis. They'll modulate to more breathing or less breathing as the feedback comes in from chemo and gas, osmoreceptors of the blood. There'll be a blood pressure regulation there that'll be getting information from the baroreceptors. And so cardiovascular, respiratory, some of those key things that we have to do to stay alive every minute of every day are going to be originating and somewhat modulated by areas in the medulla. Now, some of those areas like breathing, I can consciously overwrite and I can start making my breath bigger or less big or faster. And again, what I'm doing when I do that is I let these upper brain centers influence at the medulla the firing rate. So when I'm not letting conscious control play a role in my breathing rate, the medulla is doing it on a background kind of automation, right? So a lot of automation starts here, and then upper areas can somewhat influence and modulate. All right. Before we head to the cerebellum, let's hit the ventricles, because the ventricles of the brain are going to be embedded in the first three areas of the brain we've hit so far. So each hemisphere is going to have one big sac kind of located pretty much centrally inside. And because they're mirror images of each other, we don't give them a number of like 1, 2. So the left one is not always 1, it's not always 2. We just call it the right lateral ventricle or the left lateral ventricle, the lateral ventricles. It doesn't matter which one you label 1 and 2. All that matters is they're the lateral ventricles. They should be mirror images of each other. They should be full of cerebrospinal fluid. All right, and the choroid plexus, which is some specialized microglial cells, and then astrocytes and endothelial cells and capillaries nearby, work together to take nutrients and water and ions from the bloodstream, put them into the cerebrospinal fluid, and then pull out some of the waste products. So that way the fluid that then gets to circulate near neurons has the right balance of nutrients and availability of those nutrients to the neurons. All right, so the cerebrum, right above the thalamus, right out there in kind of the central part of each hemisphere is a right and left lateral ventricle. Now, the diencephalon, and they're going to merge on each other, and around the diencephalon, around the thalamus, hypothalamus area, we're going to see a third ventricle, and it's a little smaller, and it's going to connect to the two lateral ventricles via the interventricular foramen. Right? And so there's a little hole that lets each of the ventricles kind of feed some of their fluid into the third ventricle. And the third ventricle, again, sits somewhere, again, around that midbrain hypothalamus diencephalon region. And then we're going to see that it's going to tunnel into a smaller little ventricle around the pons and the medulla that's known as the fourth ventricle. And to connect from the third to the fourth ventricle is going to be our cerebral aqueduct. All right? So you can see the cerebral aqueduct here. And then the enlarging of the cerebral aqueduct into that fourth ventricle happens usually somewhere around the pons to the very inferior part of the spinal cord, and then it goes into the central canal. Now there's a place somewhere in here where fluid will leak into the subarachnoid space, and so that's how the cerebrospinal fluid is continuous with our meninges around the brain as well as around the spinal cord. Okay, going back to the cranial meninges, again, there's same three layers we saw in the spinal cord, the dura mater, the arachnoid mater, and the pia mater. The dura mater is going to be, again, the more thick, more like tough, collagen-rich, that's what I'm trying to say. And it's going to tie into more directly the periosteum of the bones. So unlike the spinal cord, there's not as much of a robust epidural space. So the dura and the periosteum of the bone are a lot closer to each other. Now, the arachnoid mater and the dura mater still have a little bit of space between them, so that's the subdural space. And so again, this is where Brett Michaels had his subdural hematoma. But it's not a very big space. And the arachnoid mater, again, is going to have that simple squamous epithelium that kind of leaks fluid into, along with the fluid that comes in from the ventricles that leaks into the subarachnoid space. It's going to have some collagen and elastin fibers, so it looks kind of like trabecular. It looks like it has a little bit of, you know, like tent poles holding the arachnoid and the pia mater separate from each other. And that subarachnoid space is fluid filled. And then the pia mater is physically touching and sitting on your brain matter. And so with the three meningiolators, the fluid that's present in the arachnoid mater, the little bit of maybe fluid between the arachnoid and dura mater, and the little bit of merging, and then some areas where the dura mater and the periosteum have a little space, a epidural space, that allows, again, the brain to be a little bit more protected from the skin, a little more further away from the outside world, and it's bathed in fluid, so it's not as bumping into the bone that it's encased within. The other parts of the protection, of course, are going to be that the cerebrospinal fluid's there, that we don't let blood and toxins and drugs and white blood cells come and have easy access to the neurons. We have a blood-brain barrier, and we have a very strong bone and periosteum and skin sitting, and sometimes with muscle, with the frontalis and the temporalis bones and the fascia and the hypodermis area, that helps really kind of protect. Now, because the hypodermis and because the muscle and the skin are very vascular, that's part of the reason why when you hit your head and you cut the skin or you cut into the flesh there, you do bleed a lot, okay? So cranial trauma, you do bleed a lot, and if that jolting does cause a minor little bleed in some of the capillaries that are feeding the dura mater, the arachnoid mater, you might end up with either an epidural bleed or a subdural bleed, and that's not good, the hematoma. So again, where do I make cerebrospinal fluid? I make it in the ventricles, all right? So in the ventricles, I am lining them with this myoglial or epidermal cells. And these epidermal cells, along with the blood cells and the astrocytes, are going to work together to separate the blood from ever physically coming into contact with neurons. And so in order to allow some fluid to be around the neurons, I'm thus going to take some of the nutrients that, again, from a capillary bed leak out normally, and I'm going to let the astrocytes and the epidermal cells, let them help keep in the blood most of those items that might leak out in other places, but they won't here because of the protection around them. And then what does leak out, I'm going to use the astrocytes and the epidermal cells to slowly do a little process of pulling it in and making it part of the cerebrospinal fluid, and conversely pulling out some of the molecules and taking it out. All right? So the areas where I see these specialized epidermal cells that tie into the epithelial cells of the capillaries and form this kind of unique situation of pulling items in and putting items out, that's where I'm going to call the epidermal cells my choroid plexus. And I'm going to know that the choroid plexus of my brains in my ventricles makes about 500 milliliters of cerebrospinal fluid a day. And in order to keep that cerebrospinal fluid from overaccumulating, that means later in the system I have to take out about 500 milliliters of cerebrospinal fluid a day, okay? The final part of our brain is the cerebellum, and the cerebellum is going to play a big role in gait, walking, coordination, and modulating balance. So as I stand here, it's going to help coordinate, yes, what I'm standing here doing, but also that my balance, my center of gravity, my stacking of my vertebra, my head on top of my vertebra, is engaging those muscles just enough to where I can maintain my posture. Somewhat like the spinal cord, here is the list of terms you need to know and apply to your cerebellum model and pictures for lab. And then you need to be able to describe to me what they are. So most of them are just anatomical locations. So there's an anterior lobe, a posterior lobe, and then underneath towards the anterior distal kind of downward part, there's this flocculandular lobe. The vermis is this little bit of material that kind of bumps up and is separate from the two hemispheres that runs on the center, and the axons again are running between the two hemispheres, and it's in line. The white matter is called the arbor vitae when you cut into this, and it looks like a big tree branch area. That's why it's arbor for tree, and then vitae for life. And then the gray matter, like the cerebrum, is on the outside. So if I start scratching right here and scratching a few millimeters of tissue, I'm scratching gray matter. I'm scratching non-myelinated axons. I'm scratching cell bodies and synapses and pieces of neurons that are not myelinated, dendritic surfaces. All right? And so the cerebellum cortex is the outer part, just like the cerebrum cortex is the outer part of the cerebrum. And then if you look at the neurons that live in the cerebellum, they are huge, and they have lots of dendritic branches to them. And so that's why they can be called Purkinje fibers. And they're very fast at modulating and sending information along with descending motor pathways. So I'm about to move my hand. They're very fast with their highly branched features to be able to not only help coordinate that the muscle contracts to the right amount, to the right degree, but they do it very quickly and kind of play catch up getting down there. So again, there's that flocculandular lobe, the white matter on the inside, the gray matter to the outside. When you look at how information comes in, the cerebellar peduncles are the three areas that kind of are where we start to see inner-outer movement. So the superior peduncle is the one up here, the inferior, and then the middle is more out here towards the pons. And that's how, again, the stuff coming from the midbrain and the cerebrum come in the superior, stuff from the pons comes in the middle, stuff from the brainstem comes in through the... So it's like three roads. There's three ways into the neighborhood. The superior, the middle, or the inferior road. Most of the cars coming in, most of the energy, most of the electrical potentials from the superior are coming in and out because it's going to the cerebellum. So these kind of cerebrum in the midbrain, kind of diencephalon, that area. All right, so I got to throw this in here because, again, in some books it's called the limbic lobe, and in other books it's called the limbic system. So in our book, it's the limbic system. So in our book, what they're going to say is this one gyri for the frontal, parietal, occipital, and temporal lobe that sits right around the diencephalon and right around the corpus callosum is going to be your limbic system. So it's technically from the central sulcus to this is still the parietal lobe, this is all still the frontal lobe, but this little gyri of the frontal lobe sitting next to the corpus callosum is part of the limbic system. Now, the limbic system is going to kind of have that ability to tie emotion and memory together, to like when certain smells or certain emotional triggers happen, you bring back that memory, or you retrieve like, oh, I love chocolate chip cookies and they smell wonderful, and that makes me be happy and make me think of grandma's house. All right, so that is the limbic system. And so people that have a messed up limbic systems could be serial killers because they don't have the appropriate emotional responses to things. And so this is where they're saying our limbic system, if we watch a lot of violent television, we no longer then associate violence and sadness and remorse with killing people, or because we play video games all the time and kill people all the time. So we've desensitized our limbic system. All right. So again, the three gyri that come from the frontal lobe come from right on top of the parietal lobe, and then kind of the inferior occipital and temporal areas. We can call it the cingulate, the dentate, and the parahippocampal. Now, within this one lobe here, there's this red feature known as the hippocampus. And the hippocampus is important in sleep, it's important in memory, it's important in making short-term memory turn into long-term memory. And again, having the right emotional responses with that memory is part of the reason why it's considered part of the limbic system. Okay? All right. So again, cerebrum, we have some key features to learn: frontal lobe, parietal lobe, occipital lobe, temporal lobe, limbic system. Within the limbic system, we need to know the hippocampus. We need to know the corpus callosum, the longitudinal fissure, the central sulcus, the central sulcus. The central sulcus for the frontal lobe, the very gyrus in front of it is the precentral gyrus. The outer gray matter of this precentral gyrus is going to be our primary motor cortex. The gyrus behind is going to be the postcentral gyrus, and again, the gray matter that makes the outer portion here is going to be our primary sensory cortex region. We're going to have some gyrus in front of the motor area that is going to be premotor areas. We're going to have other gyri up in the upper regions of the frontal lobe, and again, for the hemisphere, it tends to be in one. That's where emotion and personality and some higher mental functions are taking place. All right. Let's see what else. The left hemisphere is normally going to be responsible for language. So that is usually the side of the brain where our speech center, Broca's area, is going to be located. It's where reading, writing, speaking, and because the left motor cortex controls the right hand, and the majority of people are right-handed, it tends to be the dominant lobe for most people in our world, and most people who are right-handed are supposed to be good at analytical tasks, math, logic, and it usually is the dominant hemisphere. However, there are left-handed people out in the world, so that means on the right brain, their left premotor cortex is more dominant in motor control of the hand than the right hand and the left hemisphere. And then those people, even though they might be more right brain dominant in motor function with their left hand and their left foot, they still have their speech area tends to stay on the left side of the brain. Now, the left hemisphere, other areas, the frontal parietal lobe, tend to be more artistic, tend to be more about music and things. So usually people that are left-handed and right brain dominating might be more musically inclined or artistically inclined than the opposite hemisphere control. However, we all know someone who's right-handed who's super musical. I mean, there's always exceptions to every rule. And again, this is what psychology should be teaching you and talking to you about. Okay? All right, so this is just kind of a summary of the lobes and some of the key areas and associate areas that you see in the lobes. So again, our frontal lobe is going to be primary motor in the precentral gyrus, premotor planning. There's going to be a little bit of the taste that comes into the frontal lobe, but some of it's also in the temporal. And then we have a speech area, normally on the left hemisphere of the left frontal lobe. All right, the parietal lobe is where the primary sensory cortex is located for almost all of the ascending pathways, and there's 2 to 3 main ascending sensory pathways from our sensory receptors in our body to the presensory cortex here that are going to carry temperature, pain, pressure, vibration. All right, and then this is where a little bit of touch and stretch, and like, so if a mosquito lands on your arm, you know a mosquito is there, or a bug lands on your arm, you can kind of recognize that something is putting a little bit of pressure on what part of your body and then react to it, slap it or make it fly away. The temporal lobe on the outside is going to be where auditory and most of smell goes to. And then there's an associating kind of like auditory, taste, and olfactory kind of all come together. So you, so you can kind of mix, especially taste and smell come together. So that's part of the pleasure of eating and textures of eating. The occipital lobe is almost all vision. And again, the back of our brains being, if we're damaged, we might lose our sight. So some of the main specialty areas you need to know. All right, there's one big general interpretive area. Again, for most right-handed people who are left brain dominated, this is on the left hemisphere. It can show up in the right hemisphere for left-handed, right brain dominated people, but even in those folks, like Broca's center, it sometimes stays on the left hemisphere. Okay, and this general interpretive area is a little bit of everybody coming together, and it takes in smell, it takes in sight, it takes in sensory perception, motor perception, and it integrates it into reality. Okay? Speech or Broca's area, again, usually lies on the left hemisphere, right here in front of the prefrontal gyrus, the primary motor cortex, all right? And it's going to play a big role in being able to form and control your mouth, control your lips, control your throat, the muscles, and move the air so that way you can talk, okay? The eyes are going to be somewhat in this visual field that, like it shows here, So the sensory part of photoreceptors responding to light energy, that goes back here, but the muscles that control my eyes to make me look right, look left, look up, look down, you know, that part and work together as two eyeballs, that part is going to be right here, usually on the left hemisphere. Okay. Now, the prefrontal cortex, again, in front of the motor is going to tend to be a premotor planning area. And Brodmann's areas, so the original guy that mapped the brain, he gave all these little areas, so he put like little caps on our people's heads, and then he like shocked part of your brain, and because of what you said happened when he shocked like a certain area of your brain, you felt or you saw or he was able to basically kind of figure out and map the surface of the cerebrum and tell us which areas when you shock it cause light to be seen or cause movement or cause a sensation to be felt. And so he labeled all these areas a certain way, and his mapping was so amazing and so accurate that later on when we started to dig in brains and truly find out what areas guided or controlled certain things, he was right. And so his mapping and number system has carried over and actually still is found as ways to define and divvy up the brain when you do research on brains. Okay? So other key areas of the cerebrum I would like you to be comfortable with. When we start, so we've gone through all the surface, so now we have to kind of start cutting in. And so remember, the cortex, this is all that gray, all right? And that gray matter is the cortex. The white matter tends to be in the middle. Now, because we are going to have some areas in the brain where we have other synapses, and so we're going to have other unmyelinated parts of axons come together, there are going to be these other patches of gray matter that show up. And so right here you should see this is one thalamus, this is the other. The hypothalamus would technically be down here, and it kind of shows you the subthalamic nuclei, substantial nigra. Those are nucleuses, those are areas of gray matter that are associated with the hypothalamus. Well, when we come off of the diencephalon, there's these other gray matter areas that exist, all right? And some of them coordinate and play a role in the descending or the ascending pathways for sensory and motor. And so these masses of gray matter, again, are part of the way that synapses and neurons coming in or out of the brain can synapse and then trigger maybe one neuron towards the cortex, one neuron towards the cerebellum, and kind of relay information to multiple locations. So one of the big kind of organizational groups of some of these gray matter, some of these little kind of clusters of synaptic neurons and non-myelinated parts is the basal nuclei. And the basal nuclei is part of a larger functional group that kind of all together works to control some motor control. All right? And it has a caudate nucleus, which is the one kind of closest to the head. The lentiform nucleus, which I don't think they have it shown here. The claustrum, all right? And then the amygdaloid body, okay? And I think they do have the amygdala shown down here, and here's the claustrum. So this like kind of region right here, and then there, all right? So part of what this whole basal nuclei, these basal ganglia play is, again, they come into being some synaptic areas for passageways, for intercommunication between different areas of the brain. Right? And so they will play a role, and you might hear about in the basal ganglia, when dopamine is no longer being produced and being utilized by the neurons in communication, you start seeing shaking, and that is a sign that someone has Parkinson's disease. So that's kind of, when you get into the diseases, it might be that the disease is taking place with a neurotransmitter between a synapsing of one neuron and another in the caudate nucleus, or in the amygdaloid body. And because that is where it's locating and it's part of a motor pathway, it leads to the disruption of that motor pathway. And in the case of, let's say, Parkinson's disease, disruption of all that coordinating and modulating of the neural pathway means the muscles are shaking when they should be able to hold still. They're not able to do that. The muscle tone that's being maintained is not maintaining a stable, steady hand. It's maintaining a shaking hand. Okay? So just again, make sure you can identify most of these areas over here, and that you just know that while most of the major lobes of the brain and the limbic system is there, there is more to the brain and more to some of the modulating and control, and we're just scratching the surface. All right, and so some of these gray areas within the white matter of the cerebrum are known as specialized nuclei that then we've learned tie into more fine control of maybe areas that started to make their signal down towards the body from the cortex, but they get influenced by these areas. Okay. Last piece of information is you have to learn your 12 cranial nerves. So now we're moving out of the brain. So the brain is going to start to try to communicate with the outside environment. And the brain is stuck in the skull. So the way it communicates to the outside environment is it has some of its axons, some of the neurons that are tied to it have axons that leave and go to receptors in the body and take in information that way. And some of its neurons come out and exit and control muscles and other glandular and effector function. And so because the way the skull is so protective of the brain, there are only a few, major fissures and foramens and exit points for things to come in and out of the brain. So we have 12 places where neurological tissue, so neurons, axons of those neurons, they can be myelinated and unmyelinated, they kind of group together in a gaggle, and they exit out of the brain in 12 distinct locations. And where they come out and what they go and control somewhat has an ability to influence their name. So one of the ways to learn them is, of course, to learn them as cranial nerve Roman numeral. And you do need to be able to identify them by cranial nerve 1, cranial nerve 2. And they're ordered from the one that comes out the most anterior to the one that is the last one out of the brainstem. So that's how the 12 are numbered. Okay? Now, their names come from more of their function. So olfactory nerves are going to the nose to interact through the cribriform plate of the ethmoid bone and the air cells, going to interact with the receptors for smell, so chemical receptors in the nasal passageway. All right, so the neurons that are attached to those receptors are all afferent neurons. And they all are going to be tied to the receptor, axons that head into the temporal lobe. Yes, some of them will also tie into the taste centers and tie taste and smell together in the general interpretive area, but the majority of the information goes to the temporal lobe. The optic nerve, you're going to have 2 of them, and what's unique about the optic nerve is there's this point where some of the nerve crosses over, so some of what's coming from the right hemisphere does stay on the right side, and some of it crosses over, and some of it on the left goes to the left side, and some of it crosses over. And that's how you don't have holes in your vision. Now, the optic nerve is also a sensory nerve. So all of the fibers running through the optic nerve and across the optic chiasm, that crossover point for some of the fibers are going to be carrying afferent sensory information from bipolar neurons that were attached to photoreceptors, rods and cones on the retina of your eye, and that are triggered by light energy. The oculomotor nerve goes to the eye, but it doesn't worry about the visual photoreceptor response. It worries about controlling the muscles of the eye. It controls some of the reflexes of the eye. So it's part of how you know your pupil will dilate or contract. It's part of how you know to move your eyeballs in directions, right? The next one is your trochlear, and then you have the trigeminal, and then you have abducens. The trigeminal is that huge nerve for your face, so it does a lot for your face. Right? Abducens actually controls one of the muscles of the eye. So on our lab exam, this is one of the ones that you need to know, and you need to know it's the lateral rectus. Right? The trochlear innervates the supraoblique muscle. So again, it's another key muscle that that is going to control one of the 6 muscles of the eye. Okay? So when I ask you on the lab exam to identify indicated eye muscle, that's one station where there's 6 muscles. You need to be able to correctly name 3 out of the 6. When you go to the next station, it's going to be, for the indicated muscle, tell me by name or tell me by number which cranial nerve controls it. And again, I'm going to give you the lateral rectus, I'm going to give you the superior oblique, and I'm going to give you one of the other ones. And I'm going to expect you to be able to correctly tell me the 3 cranial nerves that control eye movements. All right? So continuing on into the facial is pretty important. You start to get some of your motor of the muscles of the face, and then the tongue and the cheeks, and then you go into vestibular cochlear is all sensory, getting information from the cochlea, so the sound receptors, the hair cells, as well as hair cells and otolith organs for balance and equilibrium and orientation. The glossopharyngeal gives some motor and sensory of taste. The hypoglossal, we start to talk about tongues and swallowing. Cranial accessory, we're beginning to go into the neck, and the vagus, we're actually heading towards the lungs, the heart, and controlling parasympathetic outflow. So do know name, number, function, the information it's carried, whether it's sensory, motor, mixed, and then how do I test this nerve? So I've put smelling salts up to your nose. I'm testing cranial nerve number 1. If I shine a light in your eye to see if you dilate or constrict, I'm testing cranial nerve number 3. If I ask you to read letters, I'm testing the optic nerve, cranial nerve number 2. If I do something where stand on one foot and show me if you can balance, I'm kind of testing your vestibular cochlear. If I put a sound and a wave in a headset and I ask you to tell me when you hear the beep and press a button, that's again, I'm testing your vestibular cochlear nerve. So if I ask you to stick out your tongue, if I ask you to tell me what this tastes like, again, I'm asking you for which cranial nerve. So you need to be able to tell me that. Okay, and all of that's in your lab manual, and all of that's also in tables in your book. So at this point, again, that is chapter 14, the brain, the nerves. It's a lot of fun material. It should be something, if you've had psychology, you've hopefully touched upon a little bit. And for the cranial nerves, it's cool. If you go on YouTube, you can find songs about it. You can find how to give a neurological exam that assesses the cranial nerves, and that's really cool to watch. And we used to have to do that for training in the Air Force for hyperbaric medicine before we put people in the chamber. So there's a lot of really cool things that you can do. Bring your questions to class, and I will see you next time on your video, see you next time in lab, and if you have questions, always feel free to email me. Bye, guys.